So hello everybody, welcome back to another general histology video. Today we'll be mainly talk about cytoskeleton, its structure, functions, and diagnostic applications. This will be relatively a quick video, so let's get started. So some general functions. Cytoskeleton basically consists of three basic types of fibrous proteins, and the key term is fibrous because they are insoluble. And yeah, the three types are microtubules, actin filaments, and IMF, or intermediate filaments. And they're there for support stability, basically maintaining the cell shape integrity. They create a dynamic network. So basically, there are networks of these filaments that basically can transport proteins or organelles, which you will see later on. And they are regulated by associated proteins, like how actin is regulated by tropomyosin, basically keeps actin stabilized. And some more general functions, we have mechanical, like we said, supports the cell shape integrity. Transport, they basically can move vesicles and larger molecules like mRNA around the cell, and also organelles. And we also have movement, for example, in cilia, in motile cilia, and in flagella. For example, we can see cilia in the respiratory tract and flagella in the sperm to move towards the egg. And we have cell division where, we will see that actin will play a role in that, where they create a ring, a contractile ring, so then cytokinesis is possible, which is basically cell division. And yeah, so here are basically three schemas. So three schemas of the cytoskeletons. Three schemas of the cytoskeletons. We have the microtubules, where it basically starts off from the MTOC, which is the microtubule organizing center, and they basically branch out towards the periphery. And we have the IMF, where it basically is connected between adjacent cells with the desmosomes and the hemidesmosomes. You will learn more about junctions in the next video. And we also have the actin filaments, where they are basically on the periphery of the cell, so they're mainly there for the integrity of the cell shape along with the microtubules. And they also are part of the microvilli, as you can see here. So let's get on. So microtubules. So they are relatively long hollow tubes with diameters of approximately 25 nanometers and a length up to several microns. And they consist of tubulin units, more specifically alpha and beta tubulin. And they are small globular proteins at first, but once they become to these long microtubule structures, or once they begin to polymerize, they become stable. And once they become stable, they become fibrous. And like we said, they are insoluble. So yeah, they become stabilized and insoluble. So again, they're formed by dimers of alpha and beta tubulin, and they grow from the MTOC towards the periphery of the cell, like we saw in the previous slide. And the dimers polymerize using GTP and magnesium, and they form 13 longitudinally oriented protofilaments, as you can see and here in this schema. And there is also a plus end and a minus end. The plus end is basically where the microtubule can grow, and the minus end is basically the shortening region. And the minusing end is basically where the MTOC is also located. And some general functions, like we said before, used for transportation of vesicles inside the cell, create the mitotic spindle during mitosis, and also create the inner structure of the centriole. And yeah, and so more information, how these microtubules basically transport these organelles, or basically these cell substances, is through some sort of motor that is basically used by ATP. And these basically microtubules are basically for transportation, as you can see, through a cargo, and these are done by kinins and dyneins. But the way they move is different. Kinins, for example, they move from minus towards the plus end, so basically towards the periphery of the cell, whereas dyneins move from the plus end towards the minus end, so more towards the center of the cell, towards the MTOC, basically. And so yeah, here is basically what it kind of looks like. Like, just imagine a cargo. And yeah, just more information about where microtubules can be found. Like we said before, kinocilia, flagella, cytoplasm maintains cell shape, and projections for cell migration during cell division. And here is basically just a slide of where you would find microtubules in the kinocelia and the trachea, which you will learn more about in the respiratory system, sorry, and specialized histology. And here is just another schema of um, microtubules. So now let's talk about intermediate filaments. So yeah, they are very high in tissue specificity. So depending on what type of cell, if we use some sort of imaging technique, we can find out what type of cell that is based on their type of IMF. And they can also be used in histopathological diagnosis. So in diseases, we can find what type of cell is affected using IMF. And IMF is generally found in all cells and make contact with desmosomes and hemidesmosomes, as we saw before. And they can basically arrange themselves into dimers or tetramers. And once they become some sort of like a rope shape, they become basically stabilized. You can find them in like epithelia and also in the cardiac muscles. Um, and yeah, they basically are coming in different types. Like in cytoskeleton filaments, we have epithelial cells, vimentin, Schwann cells, glial filaments, astrocytes, very important, and lamin filaments network in the nuclear membrane. Here's just another schema. We can see how the IMF is playing a part in the hemidesmosomes over here, sorry, and the desmosomes over here, which basically is a cohesion between adjacent cells. And here's just another view during electron microscope. And now let's talk about actin filaments. So they are generally about 7 or 8 nanometers thick, depending on what you read. And they consist of actin, which is the protein. Makes sense, right? And they are formed by the polymerization of G-actin monomers into double helixes. And it's also important to note that in non-muscle cells, they are unstable because they do not contain tropomyosin, tropomyosin, and troponin complex. Basically, all the associated proteins that are used to regulate the cytoskeleton of actin. And over here we have a schema of what that is, and a schema of G-actin and F-actin. F-actin is basically just polymerized G-actin. So G-actin multiplied is just F-actin. And you can see here that they become polymerized. Yeah. Some more information about actin filaments. They are associated with mobility of the cell as well, of the cell membrane as well, of anchoring more specifically. So with the zonal adherence and focal adhesions, which you will learn more about in the next video, in the next question. And they can also form contractile bundles or stress bundles, basically the contractile ring during cytokinesis. So that contractile ring will basically become like they contract itself, and that becomes smaller and smaller until that cell becomes two different cells, two different daughter cells in cell division. And they also participate in the growth of terminal parts in axons and dendrites. And like we said, they support stereocilia and microvilli. Um, and that's repeated twice, sorry. And yeah, and actin filaments and non-muscle cells are unstable because they're unstable, they go through polymerization and depolymerization. Makes sense. And so yeah, here's basically what a contractile ring is, and microvilli over here we can see found in like our intestine, for example, our brush border enzymes, which contains 30 to 3,000 to 4,000 microvilli, an important number to note because they would want you to remember that 3,000 to 4,000 microvilli, which are brush border. And we also have stereocilia, which is found in the epithelium of the ductus epididymis in the male reproductive system, which you will learn more about in reproductive system of histology of males. Here we see how actin is also playing a part in adherence, in zonal adherence, but it's also having other proteins like alpha actinin, E-cadherin, and calcium, which you will learn more about in the next question. And just to close it off, clinical applications and diagnostics. Microtubule targeting like anti-drugs can disrupt cell division and like can induce apoptosis in like rapidly dividing cells. And immunohistochemical antibodies um, in diagnostics, we can basically use that to identify tumor origins by detecting the IMF and basically revealing like the cells like tissue of origin. So basically, like before they were metastasized and their current area of the body, we can find out where it originally came from, the cancer cell. And that is basically it. And yeah, thank you for listening.